Okay, before we can continue talking about DNA and how DNA is used to make a protein, we have to talk about RNA as well. So RNA is the other nucleic acid that we'll study. Ribonucleic acid has the same structure as far as nucleotides, but instead of deoxyribose sugar, the sugar in these molecules is ribose sugar. They're smaller molecules, but they do have a similar structure to DNA. But they're not as big. They only much smaller. They have maybe 100 to potentially 10,000 bases per molecule, so much smaller. They're basically only going to be a little section of the size of the DNA. There are 3 types of RNA, and you will need to know all 3 because they each have a separate role for the protein synthesis process. And so if you know each one and what it does, then that's really going to help you understand the process of making a protein, which will be in the next video. So messenger RNA is shortened as mRNA. It has a specific job of taking the information from the DNA out of the nucleus. The next type is ribosomal RNA, or rRNA. And remember those ribosomes that were little dots on our cell pictures? Essentially, rRNA is what makes up the ribosome. So that is very important because that is where the protein is actually going to be created. And then the last type is transfer RNA. It sometimes looks kind of T-shaped, so maybe it's going to be easier for you to identify. It depends on the picture, but it transfers the amino acid to the ribosome during the protein making process. It is not a double helix. All of these types of RNA are going to be single-stranded. However, they sometimes look a little weird. So the tRNA looks like that single strand is kind of formulated into a T shape, but they are not a double helix. They do only have one strand. And again, ribose sugar. And then instead of A, C, T, and G, uracil replaces thymine. So wherever you would expect a thymine to be, you will find a uracil if we're talking about RNA. RNA functions mainly in the cytoplasm. The messenger RNA will start to form in the nucleus, and the rRNA is at the ribosome, but that ribosome is sitting in the cytoplasm. So we talked about in the last video that a gene is a segment of DNA. It carries the code for a particular protein. And again, code is kind of a weird word, but it basically is the instructions for making a particular protein. The weird thing about talking about a protein is we're going to be vague for a long time because the body has millions of proteins, but only about 20,000 genes. So the hard part about talking about proteins is we could be talking about one of a million different kinds of proteins, and they all have a different job. So the kind of protein that we're making, we're not going to get specific yet, but the process of making a protein is the same, no matter what type of protein is being made. This current definition is much more difficult. It's a little bit longer, but it basically is saying that DNA has the instructions for making RNA, and that RNA helps to make a protein, which is really essentially saying the same thing, just with more words. When we get to protein synthesis, that will make more sense. There are 2 steps. One of the steps is actually adding amino acids together. I'm sure when you learned about macromolecules that he told you that a protein is made of an amino acid chain, but the order of those amino acids is what makes a protein different. And so that sequence comes originally from the sequence of the DNA. Again, this is going to be more clear when you look at specifically protein synthesis. Again, like before in the other video, the genome is all of our genes, but it's also considered all of our DNA. And so we have 23 chromosomes, which relates to 3.1 billion nucleotide pairs. So in our DNA, we have 3.1 billion pairs making up those 23 chromosomes. That's a lot. So we know that we have 46 chromosomes in our cells. Of those 46 chromosomes, 23 came from your mom, 23 came from your dad. And when you make sex cells, which we will talk about in a few more videos from now, each of those cells will only have 23 chromosomes. It did take scientists about 13 years to figure out all of our base pairs, and that is all sequenced out, and then that information is used to identify mutations as the source of lots of different diseases and disorders that we see. The findings of that Human Genome Project figured out that Homo sapiens has fewer than 100,000 genes, and that one gene can code for lots of different proteins. It used to be thought that one gene is for one protein, but that was disproven. And then we figured out a little bit more information about how the size of a gene can be about 3,000 bases long, or up to 2.4 million. That's a big range. But the other interesting part is that all humans are at least 99.99% genetically identical. So it's crazy to see all the differences between us, but to realize that all of those differences only make up 0.01% of our genome or our DNA.